Welcome to our exploration of the tumor microenvironment and its complex interactions with the immune system. The tumor microenvironment, often abbreviated as TME, is not just a mass of cancer cells. Instead, it's a dynamic ecosystem made up of cancer cells, immune cells, blood vessels, fibroblasts, signaling molecules, and the surrounding extracellular matrix. Understanding how these components interact is crucial for unraveling how tumors grow, evade the immune system, and respond to therapies. In this video, we'll break down the key players and processes that define the relationship between tumors and the immune system, setting the stage for a deeper dive into this fascinating and vital area of cancer biology. Before we dive into the tumor microenvironment, let's quickly review the immune system. The immune system is our body's defense network, made up of various cells and molecules designed to detect and eliminate threats like bacteria, viruses, and even cancer cells. It includes innate immunity, which provides immediate but nonspecific defense, and adaptive immunity, which targets specific invaders with precision. Key players include T cells, B cells, natural killer cells, macrophages, and dendritic cells. Each has a unique role in identifying and destroying abnormal cells, including those that may become cancerous. This foundational understanding will help us appreciate how tumors can manipulate or evade immune responses within their microenvironment. Tumor microenvironment, or TME, is the immediate area surrounding a tumor. It's a complex network that includes not only cancer cells but also immune cells, blood vessels, fibroblasts, signaling molecules, and the extracellular matrix. This environment is constantly changing, influenced by both the tumor and the body's responses. The TME plays a critical role in tumor growth, metastasis, and how the tumor interacts with the immune system. By understanding the components and dynamics of the TME, researchers and clinicians can develop better strategies to target cancer and improve patient outcomes. At the heart of the tumor microenvironment are the cancer cells themselves. These cells have acquired mutations that allow them to grow uncontrollably and evade normal regulatory mechanisms. But cancer cells don't exist in isolation. They constantly interact with their surroundings, sending out signals that can recruit or suppress other cells, including those of the immune system. Understanding the behavior of cancer cells within the TME is essential for grasping how tumors develop resistance to therapies and how they can be targeted more effectively. Immune cells are a vital part of the tumor microenvironment. These include T cells, B cells, macrophages, dendritic cells, and natural killer cells. Some of these cells work to attack and destroy cancer cells, while others may be co-opted by the tumor to support its growth or suppress immune responses. The balance and activity of these immune cells within the TME can determine whether a tumor is kept in check or allowed to progress. This dynamic interplay is a key focus of current cancer research and therapy development. Blood vessels are another crucial component of the tumor microenvironment. Tumors need a blood supply to grow, so they often stimulate the formation of new blood vessels through a process called angiogenesis. These new vessels provide oxygen and nutrients to the tumor, but can also serve as pathways for cancer cells to spread to other parts of the body. The interaction between tumor cells and blood vessels is a target for certain cancer therapies, aiming to cut off the tumor's supply lines and slow its growth. Fibroblasts are specialized cells that produce the extracellular matrix, a network of proteins and molecules that provide structural support to tissues. In the tumor microenvironment, cancer-associated fibroblasts can promote tumor growth and help create a protective barrier around the tumor. The extracellular matrix itself can influence how immune cells move and function within the TME, sometimes making it harder for them to reach and attack cancer cells. Targeting fibroblasts and the extracellular matrix is an emerging area of cancer therapy. Within the tumor microenvironment, a variety of signaling molecules are constantly being produced and exchanged. These include cytokines, chemokines, and growth factors. They help regulate the behavior of both cancer and immune cells, influencing processes like cell growth, migration, and immune response. Some signaling molecules can attract immune cells to the tumor, while others may suppress their activity. Understanding these signals is key to developing therapies that can tip the balance in favor of the immune system. Immune surveillance is the process by which the immune system monitors and eliminates abnormal cells, including those that could become cancerous. In a healthy body, immune cells constantly patrol tissues looking for signs of trouble. When they detect cancer cells, they can initiate an attack to destroy them. However, tumors can develop strategies to evade this surveillance, allowing them to grow unchecked. This ongoing battle between the immune system and cancer is a central theme in tumor immunology. Tumors have evolved a variety of mechanisms to evade the immune system. They can downregulate the expression of molecules that alert immune cells, secrete immunosuppressive factors, or recruit regulatory cells that dampen immune responses. Some tumors even create a physical barrier that prevents immune cells from reaching them. These evasion tactics make it challenging for the immune system to eliminate cancer and are a major obstacle in cancer therapy. Immunoediting describes the dynamic relationship between the immune system and tumor cells. It consists of three phases: elimination, equilibrium, and escape. In the elimination phase, the immune system successfully destroys cancer cells. During equilibrium, some cancer cells survive but are kept in check. In the escape phase, cancer cells evade immune detection and grow uncontrollably. This concept helps explain why some tumors can remain dormant for years before suddenly progressing. T cells are among the most important immune cells in the fight against cancer. Cytotoxic T cells can recognize and kill cancer cells directly, while helper T cells coordinate the broader immune response. However, tumors can suppress T cell activity through various mechanisms, such as expressing checkpoint proteins that inhibit T cell function. Enhancing T-cell responses is a major goal of many immunotherapies, including checkpoint inhibitors and CAR T-cell therapy. T-cells play a key role in the adaptive immune response by producing antibodies that can recognize and neutralize cancer cells. These antibodies can mark cancer cells for destruction by other immune cells or block signals that promote tumor growth. In some cases, B cells can also present antigens to T cells, helping to coordinate the immune attack. The role of B cells in the tumor microenvironment is complex and still being actively researched. Natural killer NK cells are part of the innate immune system and can recognize and destroy cancer cells without prior sensitization. They are especially important for targeting cells that have lost normal markers, which often happens in cancer. However, tumors can develop ways to inhibit NK cell activity, making it harder for these cells to do their job. Boosting NK cell function is an area of active research in cancer immunotherapy. Macrophages are versatile immune cells that can either attack cancer cells or paradoxically support tumor growth. In the tumor microenvironment, some macrophages, known as tumor-associated macrophages (TAMs), can promote inflammation and tissue repair, but others may suppress immune responses and help tumors evade destruction. The balance between these different types of macrophages can influence the outcome of cancer progression and therapy. Dendritic cells are the primary antigen-presenting cells in the immune system. They capture antigens from cancer cells and present them to T cells, initiating a targeted immune response. However, tumors can interfere with dendritic cell function, reducing their ability to activate T cells. Strategies to enhance dendritic cell activity are being explored as potential cancer treatments. Regulatory T cells, or TREGs, are a subset of T cells that help maintain immune tolerance and prevent autoimmunity. In the tumor microenvironment, however, TREGs can suppress anti-tumor immune responses, allowing cancer cells to escape destruction. High levels of TREGs in tumors are often associated with poor prognosis. Targeting TREGs is a promising approach to boost the effectiveness of cancer immunotherapy. Myeloid-derived suppressor cells, MDSCs, are another group of immune cells that can inhibit anti-tumor responses. These cells accumulate in the tumor microenvironment and suppress the activity of T cells and natural killer cells. By doing so, they help tumors evade immune attack. Research is ongoing to find ways to block or reduce the effects of MDSCs in cancer. Cytokines are small proteins released by cells that act as messengers in the immune system. In the tumor microenvironment, cytokines can have both pro- and anti-tumor effects. Some cytokines stimulate immune responses while others suppress them or promote tumor growth. The balance of cytokines in the TME is a key factor in determining the outcome of the immune response to cancer. Chemokines are a type of signaling molecule that guide the movement of immune cells within the body. In the tumor microenvironment, chemokines can attract immune cells to the tumor or, conversely, help exclude them. Manipulating chemokine signals is a potential strategy to enhance the infiltration of anti-tumor immune cells and improve the effectiveness of immunotherapies. Growth factors are proteins that stimulate cell growth and division. In the tumor microenvironment, cancer cells and surrounding stromal cells can produce growth factors that promote tumor progression and survival. Some growth factors also influence immune cell behavior, either supporting or inhibiting anti-tumor responses. Targeting growth factor pathways is a common approach in cancer therapy. Point proteins are molecules on immune cells that act as brakes to prevent overactivation. Tumors can exploit these checkpoints, such as PD-1 and CTLA-4, to turn off immune responses against them. Checkpoint inhibitors are drugs that block these proteins, releasing the brakes and allowing the immune system to attack cancer cells more effectively. This approach has revolutionized cancer treatment in recent years. Cancer immunotherapy is a rapidly growing field that aims to harness the power of the immune system to fight cancer. Approaches include checkpoint inhibitors, CAR T-cell therapy, cancer vaccines, and cytokine therapies. These treatments are designed to boost the body's natural defenses or overcome the immunosuppressive environment created by tumors. Immunotherapy has shown remarkable success in some cancers, but challenges remain in making it effective for all patients. CAR T-cell therapy involves engineering a patient's own T cells to recognize and attack cancer cells. These modified cells are expanded in the lab and then infused back into the patient. CAR T therapy has shown impressive results in certain blood cancers, but researchers are working to adapt it for solid tumors and overcome challenges like the immunosuppressive tumor microenvironment. Cancer vaccines are designed to stimulate the immune system to recognize and attack cancer cells. Some vaccines target viruses that can cause cancer, such as HPV, while others aim to train the immune system to recognize tumor-specific antigens. Although still in development, cancer vaccines hold promise for both prevention and treatment. Cytokine therapy involves using synthetic or natural cytokines to boost the immune response against cancer. Interleukin-2 and interferons are examples of cytokines used in cancer treatment. While these therapies can be effective, they often come with significant side effects, highlighting the need for more targeted approaches. Tumor-associated antigens are molecules found on the surface of cancer cells that can be recognized by the immune system. These antigens are the targets of many immunotherapies, including cancer vaccines and CAR T-cell therapy. Identifying and targeting tumor-associated antigens is a key area of research in cancer immunology. Tumor mutational burden refers to the number of mutations present in a tumor's DNA. Tumors with a high mutational burden often produce more abnormal proteins, which can be recognized as antigens by the immune system. This makes them more likely to respond to immunotherapies, especially checkpoint inhibitors. Neoantigens are unique proteins produced by tumor-specific mutations. Because they are not found in normal tissues, neoantigens are ideal targets for personalized cancer therapies. Researchers are developing vaccines and T-cell therapies that target neoantigens, offering the potential for highly specific and effective treatments. Tumor heterogeneity refers to the diversity of cancer cells within a single tumor. This diversity can make it difficult for the immune system to recognize and eliminate all cancer cells, as different cells may express different antigens or develop resistance to therapies. Overcoming tumor heterogeneity is a major challenge in cancer treatment. Immune checkpoint inhibitors are drugs that block proteins used by cancer cells to turn off immune responses. By inhibiting checkpoints like PD-1 and CTLA-4, these drugs unleash the immune system to attack cancer. They have transformed the treatment of several cancers, but not all patients respond, and side effects can be significant. Adoptive cell transfer is a form of immunotherapy where immune cells are collected from a patient, modified or expanded in the lab, and then infused back into the patient to fight cancer. This approach can include TIL therapy, CAR T-cell therapy, and others. It offers the potential for highly personalized treatment but also presents logistical and safety challenges. Tumor microenvironment can contribute to resistance against cancer therapies, including immunotherapies. Factors such as immunosuppressive cells, abnormal blood vessels, and a dense extracellular matrix can limit the effectiveness of treatments. Overcoming these barriers is a major focus of current research. Targeting components of the tumor microenvironment, such as fibroblasts, blood vessels, or immunosuppressive cells, can enhance the effectiveness of cancer therapies. Combination approaches that address both the tumor and its microenvironment are showing promise in clinical trials and may lead to better outcomes for patients. Emerging research suggests that the gut microbiome can influence the immune response to tumors. Certain bacteria may enhance the effectiveness of immunotherapies, while others could promote resistance. Understanding the relationship between the microbiome and the tumor microenvironment is an exciting new frontier in cancer research. Metabolism of cancer cells and other cells in the tumor microenvironment can affect immune responses. Tumors often create a low-oxygen, acidic environment that can suppress immune cell function. Targeting metabolic pathways is a promising strategy to improve the effectiveness of immunotherapies. Hypoxia, or low oxygen levels, is common in the tumor microenvironment. Hypoxic conditions can promote tumor growth and help cancer cells evade the immune system. Addressing hypoxia is an important aspect of developing more effective cancer therapies. Extracellular vesicles are small particles released by cells that can carry proteins, RNA, and other molecules. In the tumor microenvironment, these vesicles facilitate communication between cancer cells and immune cells, influencing tumor progression and immune responses. Targeting extracellular vesicle pathways is a novel area of research. Chronic inflammation in the tumor microenvironment can promote cancer development and progression. Inflammatory cells and molecules can support tumor growth, suppress immune responses, and contribute to therapy resistance. Managing inflammation is an important aspect of cancer prevention and treatment. While immunotherapies can be highly effective, they can also cause side effects by overstimulating the immune system. These side effects, known as immune-related adverse events, can affect various organs and require careful management. Balancing effectiveness and safety is a key challenge in immunotherapy. Biomarkers are measurable indicators that can predict how a patient will respond to immunotherapy. Examples include PD-L1 expression, tumor mutational burden, and the presence of certain immune cells in the tumor. Identifying reliable biomarkers is essential for personalizing cancer treatment and improving outcomes. The field of tumor immunology is rapidly evolving. Researchers are exploring new ways to enhance immune responses, overcome resistance, and personalize therapies. Advances in genomics, bioinformatics, and biotechnology are driving the development of next-generation immunotherapies that could transform cancer care. Understanding the tumor microenvironment and immune interactions is not just a scientific pursuit, it has real implications for patients. Advances in immunotherapy are offering new hope, but patients also face challenges such as side effects, access to treatment, and the emotional impact of cancer. Patient-centered research and care are essential for translating scientific progress into better outcomes. Let's recap what we've learned about the interactions between the immune system and the tumor microenvironment. We've explored the key players, mechanisms of immune evasion, and the latest advances in immunotherapy. Understanding these complex relationships is crucial for developing more effective cancer treatments and improving patient outcomes. As research continues, our understanding of the tumor microenvironment and its interactions with the immune system will only deepen. New therapies and strategies are on the horizon, offering hope for more effective and personalized cancer care. By staying informed and supporting ongoing research, we can all contribute to the fight against cancer.